Thank you, Christian. Thank you very much. And thank you to Julia as well, and to Christina that have helped organize everything. And I'm very happy basically to be sharing many of these findings that we have uncovered over the past 10 years and that finally are coming together to teach us a little bit more about adaptation and how actually we had to move away from the idea of one gene, one phenotype to actually many different genes to one phenotype or correlated phenotypes, and ultimately to the connection that it might exist with speciation via the evolution of intrinsic reproductive isolation. So today you're going to hear basically two different parts to the seminar. The first one and the longest one is about polygenic adaptation in the system. And the second one is how we have found some genetic connection between that polygenic adaptation and reproductive isolation. Okay, so basically, I'm going to start with a quick background on some relevant concepts for the talk. Then I'm going to be mostly describing some of the arguments that we have been exploring to see if there's evidence for the role of natural selection in creating diversity in the system. And then I will briefly outline the system, and then I will delve into the data, and I'm going to be summarizing results from previous PhD and honor students. And finally, I'll try to bring it together how adaptation and intrinsic reproductive isolation might be genetically linked. Okay, so for me, always the main observation has been twofold. The first one is basically diversity itself, as Dobzhansky says here, but the second one is the discontinuity of that organic variation, right? So, I mean, there are gaps in between all of these different species that we observe and are fascinated by. So those two issues are dear to my heart, and I've been trying to contribute to our understanding of both. And in this system that I'm going to talk about today, we're trying to push both and also the interaction between them. So today we're going to develop some arguments for whether natural selection contributes to that diversity in terms of like forms, and whether those forms actually confer an advantage to a given environment, and trying to identify the actual trait that creates some of that advantage. And finally, whether that has some consequences for the origin of a species, right? So one of the first things that emerges from natural selection acting on a phenotype is basically that it leads to a correlation between the phenotype and the environment in which these organisms occur, right? So here's an example of many different types of what we call ecotypes in Senecio lotus, some of the ecotypes that we study in our system. But basically the message here is that you can see all of these plants have been grown in the glass house. They have a very strong genetic basis for the looks that you're seeing right now. Basically, the way to look at this correlation is that if you were to the field and collected some seeds, and you blindly gave me those seeds, and then I just grew them into the glass house, I can almost certainly tell you where they come from, and in terms of the environment. So that's the correlation that we're talking about. And natural selection explains that correlation. However, as we know, the lack of repeatability might actually say that many of these things might have occurred just by chance, and for other different demographic reasons, we might be seeing this correlation. So natural selection has this other attribute that is deterministic, and therefore we can think that as populations adapt, diverge, but if they were to do this independently and repeatedly many times, then we actually expect natural selection to lead to the same outcomes or very similar outcomes, right? And that basically is what we use as a second line of evidence for the role of natural selection. Now, the third line of evidence that we use is this variance in fitness in natural conditions, right? So for this one, we just basically are doing genetic experiments in the field where we are asking whether combinations of alleles tend to be favored more so in one environment than the other one. And you can think it in terms of like, let's say that you have 25, 50, or 75% of genes derived from this environment that you're seeing here, the sand dunes, let's say, versus in another environment where you switch the percentages. And the idea is that if you see a positive correlation between the percentage of genes in that genome from a local environment increasing, then the higher the chances of that organism to survive in that environment. So that's the other line of evidence that we try to seek in these experiments. Then finally, and this is where we're trying to get at, is trying to understand the molecular traits that underlie the phenotypic traits. So I stopped thinking about like the genotype to phenotype map in terms of like a few genes at a time, but actually like the molecular trait, which could be a couple of gene networks that interact with one another, and there might be variants in the epistatic interactions that exist between those genes, et cetera, et cetera. And we're trying to explore that via simulations and some mathematics, but we're trying to actually look at and discover the molecular traits that might underpin this trait evolution. Now, normally, if you find those genes, you actually try to refute the neutral theory in terms of its rates of molecular evolution. And of course, I mean, then you try to basically balance this burst of mutations that evolve really rapidly versus those that are evolving only in response to population size, right? So this, of course, is a simplification, but ultimately we will have to deal with these, explaining these patterns. And whether we can explain these patterns under simple models of one or two loci versus more complex traits where hundreds of loci might contribute to the trait change, then that's something that we're also quite interested in. All right, so all of this, of course, it doesn't live in a vacuum. Of course, I mean, it lives in a body of theory that has been developed over a long time, and many of you in the audience have contributed to the development of this. And in general, I mean, the way we think about it is we look at adaptation in multivariate space in which we have multiple traits kind of like evolving towards an optimum that in this case is highlighted in the middle of this hypervolume. That is the optimum. And that red line is basically as how the population is depicted to approach that optimum. Of course, I mean, you can think of that in terms of serial mutations that are adaptive, or in polygenic circumstances, you can think of that as the trait moving as many alleles shift their frequencies as well. Now, the approach to this optimum, we think of them in terms of diminishing returns. And as you know, like in a microscope, you have a big dial and a small dial. So you can think that at the beginning of adaptation, big dials might be moved and you try to focus that image, but then you need to use smaller dials and then you can do more and more and more until you truly are hovering around that optimum, and every new mutation basically is kind of diminishing returns on top of the fitness gains. So overall, I mean, the way we think it is that many of the mutations that are adaptive are common, but small effect, but there will be some rare of large effect that might be favored, particularly if you are very far away from the optimum. So all of these things are in our mind as we actually try to think about adaptation in this system. So, so these two plans that I want to talk about are the dune and the headland, and I'll tell you about them in a second, so we can actually think about them in greater detail. But we've been trying to develop multiple lines of evidence, first for whether these plants have been evolving by natural selection, right? And once we have sought enough evidence and found enough evidence to demonstrate that, then we have switched into finding the actual traits that might be evolving in their selection, and then we're doing the genetic architecture of those traits, right? So as you will see, my talk will be mostly about adaptation because that is really hard to demonstrate, and here we're going to show you our approach, basically. And then finally, we're going to see what we have found in terms of its connection to reproductive isolation, okay? So now I'm going to switch gears to providing some background on the system of study and how these different lines of evidence have been produced over the years, right? So the genus Senecio is a hyperspecies genus that is found worldwide. There are pretty much any type of growth habit, polyploidy series, life history, trait variation, flowers with petals and without petals. I mean, you name it. It's incredibly specious. And one of the things that I love the most is that it's taxonomically a mess. And that for me highlighted at the beginning that this conflict to taxonomically trying to decide what is what suggests that many of these populations might be at flux, and it might be carrying a lot of ancestral polymorphism. So many of these things might be in the very young type of radiations. So this genus has been studied in Europe quite a lot. For example, Richard Abbott, John Brillen and others, and Simon Hiscock have tried to explore hybrid speciation, and this genus is mostly well known for that. But here in Australia, we have started to develop a new system within this genus, and that's basically what I'm going to be talking about today. I showed you before some of the variation that we see in this taxonomic species that we call Senecio lotus. There's a lot of a fight between New Zealand and Australia in terms of like trying to figure out where is lotus, whether in Australia or New Zealand or both. And many of them consist of a bunch of taxonomic species that are called, for example, Senecio pinnatifolius, Senecio brigaloensis, and so on and so forth. I tend to avoid that, and I've been trying to look at the overall diversity of the entire species complex. And that's why I'll keep referring to it as the Senecio lotus species complex. The two protagonists of today's talk are the dune and the headland ecotypes. And these ecotypes occur by the coast, and they punctuate the coast every 100 to 200 kilometers. And every time they occur next to one another in parapatry. And this parapatry sometimes can be a matter of a couple of meters or 3 meters, which is basically the four-wheel drive road by this beach to get the cars inside the beach. And then you can see like the rocks and the sand dunes next to one another, basically, and a small hybrid zone in some cases, which might be a few meters only. It's a very strong selection against hybridization. Also, you can find them separated by 1 kilometer, like it might happen in places like Lennox Head. And what we believe is that, I mean, a lot of towns, you might imagine, have been built in these beautiful places. So I think that they have actually broken many of these populations. But anyway, that's for another story. So this coastal ecotypes, the headland is the one that forms those cushions on top of the rock. You can imagine that the soil bed in those headlands is very, very thin. So it's very tough for those plants to basically shoot root deep into the ground. Difference to the dunes on your right, which actually have a tall phenotype, and then the roots actually go down seeking for water. The headlands are very hardy plants. They deal well with salt and with drought, as opposed to the sand dunes that require more care. And their architectures are slightly different, etc. And I'm going to show you some of the data so you can actually picture in your heads how these different populations look like, okay? So here we have a map on the right where we have sampled many of these dune headland populations. We've sampled them on the eastern coast and the southern coast. In the western coast, we haven't sampled pairs because there's only one known pair in an island and it's actually protected, so we cannot actually study it. But on the eastern and southern coast, we have plenty of pairs to ask questions about them. And on the left, you see many panels. I wanted to focus on the top left panel that has an asterisk. And that one shows lines that connect a dune population with a headland population, with its pair. And as you can see, like in that case, we're looking at vegetative height. The dune of the pair is always taller than the headland in that pair. And that happens to other traits. For example, in the right smack in the middle of the graph, you have secondary branch angle. And you can see there that is a striking trait that is highly correlated with the environment in which they exist. And that happens in average for many traits. And when you look at that in multivariate space, and you look at all the traits at once, you can see in the center the PCA of all of that trait combined. And you can see that the dune and the headland sort of like form these two big clouds. And that in and of itself vouch again for the correlation between the environment and the morphologies. However, this type of association across many pairs could have resulted just from one single origin and then trickling down the coast and colonizing similar habitats. But what we have found, my student Mary James found that many of these pairs are actually colocalizing with one another in phylogeny according to locality, not according to pair. We have seen this in at least 3 independent studies that we've done, in which we have looked at phylogenetic relationships across these populations. This one is particularly robust because we have around between 60 and 100 individuals per population, and we have thousands and thousands of markers that make us very confident about the relationships that we see in these populations. These populations are also very isolated by distance, and many of the populations that are close to one another are isolated to the extent that levels of gene flow are almost negligible. Right, so based on some simulations that we've done and some estimates of isolation with migration models, we have basically discovered that these populations are pretty much independent from one another. Any headland from any other headland, and dunes less so, but still independent from one another. And in multivariate space and in environmental space, the dunes are very similar. I mean, it's like a much more homogeneous pool. The headlands are much more heterogeneous, both in environment, in the soil, for example, and some of the morphology, but they are definitely prostrate, or these form these cushions on the ground. Now, these populations, in the multiple transplant experiments that we have carried out in the field, they show consistency in the direction of local adaptation, both across sites and time. So this is an example of 4 transplant experiments that we've done at the same location from 2012 to 2016. And even though selection fluctuates in strength over time, what we see is that the direction of selection seems to be always the same. In 2013, we can see how the distance between the survivorship differences are not so much, but then we have 2014 being greater differences in 2012 and so on and so forth. So we believe that these populations are being pulled apart in these environments consistently, okay? And I'm not showing the data here, but we also have the same for other population pairs in other localities where we have the same signatures. So overall, what we find is that this system has evolved in parallel many times, these dune headland pairs that are strongly locally adapted to their environment, okay? So that suggests that we have at hand a system whose some of the traits might have evolved under natural selection. And in 2013, or well, 12, because this was published in 2013, in 2012, my student Federico started to look into what is similar between the pairs, right? So we look at divergence in each one of these pairs on the map on the left, and we ask, okay, well, are they divergent in the same or different? And at this time, we were pretty much into the paradigm, oh, we're going to find one gene explaining the whole thing, or a huge amount of the variance, but we didn't. Basically, what we found is that there were many loci different between each pair, each population and each pair. But when you compare pair 1 versus pair 2 versus pair 3, as many of you have actually witnessed in your own research there in Vienna and in other places, we just couldn't find anything remotely similar across the pairs, except a few loci. And those few loci are highlighted in the bottom neighbor joining phylogenies, where you can see in the left is just all the loci, and it shows the pairing by locality. Both the orange and the green come together by pair. But then on the right, panel E and F, you can see that we can separate with these loci. It's not too many of them. If we plot the frequency of all of these loci into a neighbor joining tree, what we see is that we can separate the dune and the headlands, right? So that hinted to us that, well, it's frequency which might be shifting all of these, because there was no single locus per se at the time that would separate the two types fully, like as it might happen in sticklebacks with the gene ERA. So we looked at, okay, but what is similar then to these genes? And what we found is that some types of predicted functions in these genes was repeatedly divergent across these pairs, but not the same genes. So I just want to point out to the table on the right, the purple bit, and there are some numbers on the third column from left to right, and it's 8, 7, 7, 7, etc. And it keeps going like that. That is the number of pairs in which there's an enrichment for genes that contain SNPs in this type of categorical functions, right? And again, they are predicted ones, so we're been using that to generate hypotheses to test, okay, if this is a polygenic architecture of adaptation, well, how are we going to get about it, right? And that's basically what I'm going to basically talk about for the next bit of the talk. So far, I've told you that we have parallel evolution in a system where there's a strong correlation between environment and phenotype. And we see that both at the univariate and multivariate level. And we know that there's a strong local adaptation in these pairs. Now, we have also seen that these pairs differ genetically from one another. And it seems that at the SNP level, there's very little to say about how common their divergence has been. However, when we consider like how these genes contribute to a pathway or to predicted functions, then we can actually explain way better the patterns of convergence that we see across these different pairs. So with that in mind, I'm going to basically show you here how is that we're studying this system, this molecular basis of polygenic adaptation, right? And that there are multiple systems across plants and animals that have been at the forefront of doing this. And of course, you're privileged to have access to these phenomenal systems that from Drosophila that have been developed there in Vienna that allow you to do experimental evolution to the extent that we could never do it. We do it in a different way. So I'm going to show you how is that we do it, okay? And just what we do is, as I said, we generate, if I can get rid of that little screen, so that I cannot see my mouse. If anybody can, if you are seeing the screen where I am, etc., and it's covering something, if you can move it out, that's great. If not, no problem. It's just that I don't know what you guys are seeing. All right, so as I said, we have this population genetics signal, right? That says, look, I mean, there's a lot of differentiation in these pairs, and this differentiation can be explained in terms of like pathways being differentiated, or these molecular traits that I've been calling, and then say, okay, well, so what can we follow that up? So one of the traits, molecular traits that was consistently enriched in the differentiation of these pairs was signaling by hormonal plants. And these hormonal plants, like for example, auxin, which I'm going to talk about it in a second, I mean, they are incredibly powerful. And you must have possibly heard that auxin controls everything. And it's true. I mean, auxin affects all sort of patterns of growth and development in plants. And so normally it's been like, oh, whatever. I mean, like auxin, that's not informative, right? And but I mean, I'm originally trained as a geneticist in Drosophila. I've started to become an evolutionary geneticist in plants over the 10 years. So I had the advantage of being really naive about plants themselves. And what we decided to do is ask, okay, so we have this auxin pathway that has been studied to death in Arabidopsis, let's say. But how is that they have found the genes involved in that auxin pathway? And what they do is they do mutant screens, right? So they get this population of Arabidopsis, and then they blow it up in the population while adding some chemicals. And then the single gene mutants, then they look at their phenotypes, and then they discover the gene that controls that phenotype. And that's the way they have put together these pieces of the clock, so to speak. And this aim of developmental biology is incredibly powerful. And what we ask basically is, what are those phenotypes? So we say, okay, every time that they have mutated the auxin pathway, how do plants look like? Okay, and there are a bunch of phenotypes, of course, that emerge. But there was one of them that caught our attention, and is that auxin control plant movement. And maybe not surprisingly, but Darwin actually wrote a book on the movement of plants. And Darwin was the first one to actually suggest that there might be something that controls the movement of plants in response to light cues or gravity cues, etc. So we asked, can we find these phenotypes, these mutant phenotypes in nature? We would have never thought of actually look for them because we often measure the plants when they are adults, etc. But we ended up doing Petri dish experiments like in Arabidopsis, and we focus on these molecular traits controlled by auxin that had to do with the movement of plants. And it's because if you talk to the developmental biologists down the corridor and you tell them, oh, look, we found auxin divergence in this tall and short plant. And they will say, oh, yeah, of course, there's nothing surprising about that. And I'm like, what do you mean? And they say, yeah, that must be auxin gradients doing it. And the reason they say it is because auxin is known to actually migrate from one side of the stem to another side of the stem, proliferate cells there very fast, and because physically this other side of the stem is not actually growing at the same speed, then the plant bends, right? So this happens if you put light to the side, but it also happens if you actually turn the plant on its side, as we're gonna see. So overall, what we think is that auxin drives these tropisms, and what we're thinking is that the tropism response controls whether you are cross-straight or you are tall in these plants, okay? So there's one type of tropism called gravitropism. You have like the vector of gravity going from up down, and plants have negative gravitropism. They grow against the vector of gravity, yeah, and against the pull of gravity. So that suggests that if you put the plant on its side, that is shown here, then the plant will align itself with the vector of gravity and will shoot up, okay? The roots also have something like that, but we're focusing on the shoot gravitropism, right? So we tested that hypothesis that this trait existed and that it was different between the dune and the headland. So the prediction is that the dune types will be a gravitropic, sorry. So when you turn it on its side, the plant will actually go up, as shown in the left, and the gravitropic ones, the headlands, will not. Or if they do, will be slightly less. So we can measure the angle, basically, to which they turn. And this is the data by the work from Melanie Wilkinson, who also did her PhD here. And here at the top in purple, you can see the difference in gravitropism between the dune and the headlands. And in 1, 2, 3, 4 of the pairs that have very strong differences in height, we have a correlated response on the gravitropism. And in those on the right, millicent, and another one that doesn't have differences in height as much, then the differences in gravitropism are actually not significant, or sometimes even in the opposite direction, right? We have one interesting one at the very left of the panel B, Stradbroke Island, that is mesmerizing because it's the only one that has incredibly differences in height, but gravitropism doesn't respond at all. So it's possible that there might be even other solutions. But overall, this gravitropism, we're starting to get a hint that there's again a correlation with environment and evolve in parallel. But the interesting thing is that when you look at height and gravitropism across all those natural populations, you can actually predict the gravitropic response of a population where you measure height, right? But that only works if you actually separate the populations by the two clades, the populations that are in the eastern coast and the populations that are in the southern coast. If you remove that information, you would imagine here in this graph, there would be just like a cloud of dots that wouldn't say absolutely anything. So one of the benefits from having sampled so broadly is that we were able to actually discover that there are two major radiations, basically, one in the east and one in the south, for the evolution of these traits. Right. So it seems that everything is hinting at natural selection is driving also gravitropism, and that gravitropism is correlated with height and other auxin-related traits. So how do we actually show that gravitropism is actually under selection? So here's where quantitative genetics came to the rescue. And what we did is to create a recombinant population where we shuffled all the genome of the dune and the headland population. We took like 30 different parents from each. They're highly heterozygous populations. And therefore, we create 3 replicas of those populations moving forward to 8 generations of random mating equal contribution. So we keep the variance constant through time. We remove some of the effects from drift, but we definitely don't undergo any selection. And then what we do is we take that F8 populations and we put them in the field, in the sand dunes and in the rocky headlands, and we did 3 rounds of selection in the field, right? And because in plants we can keep the seeds in the fridge, we always have a family from which we have 50% we kept in the lab and 50% goes into the field. So we always have even the seeds for the plants that died for those families, right? So we can actually compare always fitness between survivors and non-survivors back in the glasshouse. So what we did is that after 3 rounds of selection, Melanie took 1,200 plants or more and measured gravitropism in all of them, okay? And what she did is that, okay, well, what happens to the fitness of those plants if they come from a plant that survived well in the dune, and what happens to the phenotype of these plants if they come from a plant that survived very poorly in the dunes, right? So you can imagine they were looking at the response of the trait, yeah, on the offspring from the plants in the field that survived well or bad in the field. So I'm going to show you in this graph. On the y-axis, you have the offspring trait value. And in the x-axis, you have the fitness value that we measure in the wild, okay? And what we're asking is, in the left side of the panel, you're going to see the dune environment, and on the right side of the panel, the headland environment. And what you're going to see is we are answering the question, families that had very low fitness in the wild, what is the phenotype that they will produce in the offspring with respect to gravitropism? And families that were really good at surviving in the wild, what is the gravitropic value of them when we grow them here in the greenhouse? And at the top of the scale is gravitropic plants, and at the bottom is not gravitropic plants. And this is basically what we see. In the dune, there's a very strong correlation between the fitness of the plant in the wild and the offspring that they would produce with respect to gravitropism. And it's in the expected direction, like high fitness plants are expected to become gravitropic in the dunes. In the headlands, we seem to have removed a lot of the variation, but as you can see, like the maximum value in the dune is above the average value on the headland. So in the headland, it's still unresolved for us why exactly this happened, but there seems to be an average response that is actually low relative to the highest fitness in the dunes. But overall, we know that in the dunes, at least, the variation that was aggregating in the field over 3 generations, that variation in fitness co-varies with the variation in gravitropism. So it seems that in the gravitropic value is also favored by natural selection in these environments. But remember, the gravitropic value reflects divergence in the molecular trait, right? Which we predicted would exist based on population genetics differentiation. Right? I mean, some people might have actually studied gravitropism because they love gravitropism, but we studied gravitropism because it was just a predicted trait, phenotype, based on the divergence of a molecular trait that seemed to be divergent between populations, okay? So now what? So what do we do with it? So there's still one major problem. In the recombinant populations, we are desegregating, decoupling many traits from gravitropism, right? So we might have the time to flowering. Well, we know that it's earlier in the headlands than in the dunes, right? So that's another divergent difference. And well, maybe they are correlated or not. So in the F11 plant populations, we are actually letting segregate independently whatever traits are actually uncorrelated. So that, when you compare it to natural populations, is a problem because we can only see differences between the parental populations, right? So we have the problem of divergence in the molecular trait and the genetic architecture of the divergent trait. So we're going to try to decouple the two, right? So this is our molecular trait. It's called the auxin pathway, and also the response to gravitropism itself. It's very complex. I'm not going to run you through all of this. Just to say at the top, these different categories, biosynthesis, conjugation, and so on and so forth. Each one of them contains different number of genes that have been shown to contribute to those steps in Arabidopsis. Yeah, that's where functionally they have been procured. In our case, we are actually annotating our genes in Senecio based on this information for the best orthologs that we have. So we are conservative in terms of saying, like, once we find one of these genes, it's a good candidate to keep pursuing, right? So what we did is, on the first hand, we took the F11 population and we took the tails of the distribution, highly gravitropic and not so much gravitropic, yeah? And we sequenced 140 genomes of them. We have a genome draft. It's not perfect, but it allows us to actually look at the codependency of different genes and get intronic predicted regions and exonic predicted regions. So we sequenced around 70 genomes in each one of the tails, and the effect size is massive. I mean, like, we're dealing with, on average, 50 degrees in change, which is one of very large effects. So we're banking on having we banked on having a high power based on increasing the effect size, and by having enough replicates within each one of the tails to actually calculate frequencies of alleles, because these frequencies would reflect the frequencies that we see in nature, right? This is a population that is possibly derived from a good random sample of the variability in nature. Most likely will be smaller, but at least it's a good sample. Now, the other thing to consider here is that this F11 population, again, is segregating different traits. So when we take the random F11 and sort them by trait, we are effectively imposing incredibly strong selection, right? And we're saying, okay, what happened to their genomes? What got reconstituted, if at all, right? So we compare that to the 80 genomes that we sequence in some of these populations that we have sampled so far. Right now we have around 300 genomes, but I'm just going to show you the first indication of what is going on. Okay, so let's understand this graph first. The first three panels, right, are the data from the natural populations. And the bottom panel is the data from the F11 once it's sorted by the gravitropic tail. And what we are measuring is the allelic frequency difference between either the two tails or between a dune and headland pair when we have three of them. Now, the gray regions are exons and the white regions are predicted introns. And well, this gene, which is a auxin transporter, as you can see, the pattern of the three pairs is incredibly similar, right? And which is mesmerizing by in and of itself. But what is actually remarkable is that if you take the F11, which if you take random samples from them, you wouldn't see any difference in allelic frequency between those random samples, because the average allelic frequency between those random samples is 0, and the range is no more than 0.2, right? In here, once you sort them by the trait, you recover, it's uncanny, the same divergence that we saw in nature, right? And some of them are intronic, some of them are exonic, but the main peak of differentiation is an intronic section, which is known to contain also some transcription elements that are bound by other proteins, right? So here we have a great candidate to follow up, but we also have other type of signals, which we find very interesting. And the top three panels, again, the natural populations, and then you can see patterns of divergence across the gene. In the main intron in the middle, there's a lot of differentiation. And then in the exon 3, there's also some differentiation in one of the populations, the one in Lennox head, number 3, from top to bottom. And what you see is that once you sort in the F11, you get rid of all the intronic divergence, right? Which means that we desegregate it, decouple it, but you retain that signal in the exon number 3, which is derived from the population Lennox head. That F11 is derived from the population Lennox head. So we believe that we have another nice signature that is candidate for explaining this differentiation between the dune and headland at the gravitropic trait. And finally, we have another striking result, which is most likely a long haplotype of high differentiation between the dune and the headland across the three pairs. But once you let that segregate in the F11, you remove all the differentiation except for a region right where a predicted transcription binding factor element exists, right? So when I say that we're decoupling divergence from genetic architecture, this is exactly what I'm saying. If I were just only looking at the natural populations in this gene, I would not be able to say pretty much anything, right? It would be really hard to figure out what is going on. Maybe I would say, oh, maybe the gene is involved. But it was the F11 population, and comparing to the patterns of divergence in nature, that truly highlighted what is going on here. Right, so overall, we have some genes for which we know based on our Gravitropism what they do, so we can actually now create physiological predictions as to what we should see according to the genotypes that they have, right? And that has basically is triggering a lot of our research at the moment. Right, so far I told you how we have approached our idea of this molecular basis of a polygenic trait by actually taking advantage of these predicted phenotypes should a molecular trait be mutated, right? Now, the final question is, well, I mean, we know that we have a trait that is likely evolving by natural selection or is strongly correlated to another trait, but it seems that the biology of the trait is consistent with the idea of explaining at least the growth habit of the plant, which makes sense that is adaptive in those environments. But I mean, what are the consequences of this for discontinuity? Is just adaptation and just evolution of forms and that's it? Well, I mean, we start to actually now shift our efforts to looking at the origin of discontinuity, these gaps that exist in the distribution of these phenotypes. So I told you before that we have these two clades, the southeastern clade and the southern clade, or sometimes we call it eastern versus southern clades. And what we did is basically something very simple. We did crosses between clades and within clades, yeah? And the interesting thing is that by doing it between clades and within clades, we have population, which is a random effect here, but then we are correcting by divergence. All of the crosses are the same divergence, which is around 400 to 500,000 years of divergence, right? So we do crosses within and between, and the predictions are very simple. Crosses between clades or within clades of the same ecotype, they produce a lot of seed, but the ones that are between clades or within clades of different ecotypes, they should produce less seed, okay? And this is what we see. So on the left, we have a seed set, and on the right, we have viability. And the results are quite similar. Now, what I want you to pay attention to is that when we have different, same clades, right, crosses within the same clade, what we see is that the average seed set and the average viability is pretty much the same whether you are crossing the same ecotypes or the different ecotypes. For seed set, there's some variance, which is quite interesting, but there's no trend. It occurs both for the dune and the headlands. And sometimes they even have hybrid vigor. You cross them and they produce even more seeds than the parent. Yeah. Now, the interesting thing is what happens between clades. So when you look between clades and you cross different ecotypes, they produce very few seeds, right? That's on the left panel at the top. Reproductive isolation is almost complete, right? Very hard to produce seeds. And for viability, well, I mean, they're still suffering. They just don't produce as many plants that live all their lives. But the interesting thing is what happens when you cross the same ecotype. So on the left, if you look at the orange dots, the average crossability and viability is actually as good as the parents. Even sometimes they have hybrid vigor. 500,000 years of divergence, and you can easily cross them within ecotype if they are from the dunes. But if you cross the headlands, right, they have great difficulty in producing offspring, viable offspring, and mostly fecund offspring. So we think that as these headlands have originated, which we believe is the derived ecotype, all plants are tall in these genus, every time they have reached a solution, a polygenic solution for adaptation, they have evolved these different solutions that might actually lead to incompatibility. So we say, well, what happens therefore if we have this adaptive trait and we do crosses now between the gravitropic individuals and the agravitropic individuals from the F11? Do we find evidence of a genetic correlation between fitness and the phenotype as we do the crosses within and between tails, in this case, which would mirror the crosses within and between ecotypes? And this is what we see sorry, let me just, and I'm going to show you what we see. Now, one other piece of evidence that is important for us to justify this approach is that one of the candidate genes that explain gravitropism is one gene called enodal that is an opsin transporter. And this enodal gene controls gravitropism to a certain degree, and when it's interacting with another gene, ABA3, which is a gene that has to do with resistance to stress, salinity and drought, etc. Then when you have that and you combine the double homozygote genotype for the two genes, then you get a much greater gravitropic response or a weak gravitropic response that would be in the headland likes. So at the right, you see the CCGG genotype plants. They don't respond to gravity. They don't bend as gravity pulls. But the other genotypes, they just behave as the parentals, basically. Yeah. Well, what is interesting about this gene is that the gene was not discovered with respect to shoot gravitropism, but it actually was discovered with respect to pollen tube reception, right? So this gene is pleiotropic, effectively, for gravitropism and for the ability to pollen to actually be received by the pistil of the plant as it moves through. So we say, okay, well, we definitely have to test this hypothesis that gravitropism is related to covariant, it's covariant with fitness. So we did this process I mentioned, within and between the tails. And when you look at within the agravitropic tail, there's no failure at all. You can produce all the offspring. When you look at within the gravitropic tail, 5% failure, okay? But when you look between the tails, there is 21 failure of the crosses between them, right? So somehow the genes that have been increasing frequency by ourselves, by putting them in that tail and in the other tail, the increase of those frequencies in those genes have led to some form of incompatibility. And this is intrinsic because it has nothing to do with the environment in which we grow them, right? Which is also true when we do experiments in the field. But in this case, it happens under any condition, and there must be some underlying connection at the genetic level that links basically the polygenic architecture of the trait with the epistatic failure that occurs as you cross two groups, right? So now we're actually in our aim is to test the genetic architecture of this failure rate, and a new PhD student, Brianna Goff, is actually looking at isolating potential Dobzhansky-Miller incompatibilities within these tails, and actually see whether that model applies to the standard view of the evolution of intrinsic postzygotic isolation, or whether we need to revise how this isolation evolves. So we're also doing a lot of simulations to recreate this type of patterns, right? So overall, we think that there might be a covariance, genetic covariance between the two traits, but the actual mechanism that explains how intrinsic reproductive isolation and polygenic adaptation happening, it's still unclear to us, but it's the focus of our research at the moment. All right, so overall, I've shown you that we have a system where many lines of evidence suggest that natural selection is driving its evolution, and particularly this pair of plants, the dune and the headland, seem to have evolved repeatedly and independently their morphologies as they have colonized these extreme environments along the coast. And we know that they vary in fitness in the wild, and is related to the proportion of genes that they carry from a given environment. And we also know that these plants, we can segregate a lot of the variation in fitness into traits, but the way we have done that is by actually looking at population genetic differences between them and creating hypotheses for molecular traits that should be divergent. And once we find these molecular traits, then we ask, okay, what sort of phenotypes are predicted to emerge should these genes be mutated? And that's the way we have created these predictions for the gravitropic trait, for which we also demonstrated that it's evolving by selection in parallel and in response to selection in the field in the expected direction, right? And then finally, we show you how the combination of the polygenic architecture of the trait and the patterns of divergence, we can combine them to disentangle divergence in the molecular trait per se versus divergence in the genes of the molecular trait that contribute to the differentiation in the adaptive trait, right? And that's basically what we did until now. And finally, we said, well, look, there seems to be a genetic correlation between the trait variation and fitness variation according to where the trait that the populations carry, and also according to the environment in which they exist, right? So it seems that ecology and adaptation to local conditions is driving the evolution of intrinsic postzygotic isolation as well. All right, so of course, none of this would have been possible by a group of highly talented and wonderful students. My lab has been comprised of PhD students until now. We have the first postdocs joining our lab now, and by the way, we are recruiting, so if anybody's interested in joining and help us discover more of this type of mysteries about diversity and speciations, please get in touch with me. But them are the responsible for discovering all of this. And this is just a fraction of the work that I've showed, and I wish I could do more justice to them in all of the things that they have done over these years. And also, this has been possible because of my family. We are in very tough times, and we need to take care of each other, and we need to basically move together as much as we can, both with our relatives, but also with our friends, and with the people that we have down the corridor, you know, and the people that, well, used to travel with us and is day to day next to us. It's very important to make this sustainable. And of course, I mean, I want to thank you for coming and listening to this talk. And again, the PopGen Vienna Graduate School of Population Genetics for the invitation. Christian, thank you very much for inviting me, and to Julia and Christina for organizing everything. And with that, I'll finish, and I'm very happy to answer any questions you may have.